So welcome everyone to our roundtable that we have entitled Data-Driven Approaches in Healthcare and Precision Medicine, Challenges and Opportunities. This roundtable closes the 3 training days you had at AI4Health Winter School before. Let me first present our speakers. We will turn their camera on. So we have Dr. Billy Amzal, who holds a degree from École Polytechnique and a PhD in Bayesian optimal designs. He has worked on model-based drug development at Novartis, and now is the CEO of Quinton Health. And he has a lot of interest in real-world evidence. We have with us Professor Jim Collins, Professor of Medical Engineering at the MIT. He works a lot on synthetic biology, and we'll talk about AI-driven drug discovery. We have Professor Christian Ohmann, formerly from Heinrich Heine University Düsseldorf. Now he works at ICRIN, the European Clinical Trial Support Infrastructure, where he leads, among other European funded projects, SimCore on in silico trials. We have Professor Kitrus from Radboud University Medical Center in Nijmegen in the Netherlands, who has interest in innovative designs for clinical trials, but also works on bridging the gap between real-world evidence and clinical trials. Kit is also the chairman of the Biostatistics Working Party at EMA, and Dr. Ramzi Temani, who is the scientific director and head of France AI and genomics at Janssen. He holds a PhD in bioinformatics. He has worked a lot in academics and in the pharma industry, and he has a strong focus on bioinformatics and genomics. Thanks to all of you to talk to this panel. And I would propose to start with Billy Amzal, if he agrees, to give us a first short lecture. Sorry, I forgot to tell you that first we will have a small talk by all our panelists. Then we will have questions and answers. Then we will have some time to take questions from the audience. So please do not hesitate to ask a question in the chat, and we will pick up some of the questions, hopefully the ones who look to get more interest. But anyway, feel free to ask questions, and the panelists will react to your questions. So Billy, the floor is yours.
Thank you, Raphael, and thank you all for joining this discussion on AI-driven decision in healthcare. And here I will talk beyond R&D and beyond discovery. So, and my a little bit provocative title is can we simulate the real world? I will be short and focused, but I will just basically initiate this thinking process and the discussion around it. So maybe I will start with a very common example, very familiar example around COVID. Sorry, that would be my only 1 minute on this topic, but you have realized how this role of predictive analytics was instrumental, not only for drug discovery or trial designs, but really for public health decision making and very impactful decision making. You see, those typical picture. I don't know if you can see my pointer, but those typical modeling outputs were really, broadcasted every day, at least in France, on the news. And our prime minister, our president were commenting on those modeling outputs. Just that's, typical and very illustrating on the impact of those modeling tools, and really how they could be useful for decision making at large scale. So of course on public health, but also more generally speaking on impactful business decision making, again, beyond discovery. And of course, when it comes to decision making at a large scale and very impactful decision making, you are using all the data available or relevant data. And then, of course, you are facing multiple and heterogeneous data sources that you want to integrate ultimately in one common analytical stream. And data types may range from, of course, RCTs, RCT data, patient level or aggregated data from the literature, for example. You may have subgroup data as well. You may have, of course, real world data from care, so from electronic medical records, from claims data, from surveys. And so you can see the variety of data sources, and of course they come with different limitations, different depth, different gaps, of course, and they inform different parameters or different aspects of a disease or of an exposure to a virus or to a treatment. So you can inform, for example, drivers of an exposure or drivers of outcomes. You can inform how patients are switched from a treatment to another. You can inform how disease is progressing over time. So you can inform, for example, a score over time, or you can inform long-term outcomes like, cardiovascular risks, for example, or overall survival, and so on and so forth. So now, when it comes to decide, you need really to integrate this data into one stream, one integrated evidence platform. And that's my point I want to make here is really to kind of integrate also the analytical tools that we have in hand. So for subgroup identification, for sequence mining, for NLP, to extract data from literature, and from also classification algorithms, causal inference, to put that all together into one unique decision-centric platform, so an analytical platform or modeling simulation platform, to really integrate this evidence and be able to inform basically decision making. So that's the whole idea. And I think that's where all the players from decision making side, but also from the academia or from industry, that's I think one ultimate goal that we all need together to work on and to make it happen, in order to ultimately make the personalized medicine, but also the data-driven healthcare, as we say, possible. And I think in this perspective, what you can extract from all those data and from all those analytical tools is to inform conjointly both the real-world disease progression, meaning to characterize progressor profiles, to characterize how disease is progressing over time, to be able to diagnose patients earlier and so on in real world, and on the other side, to inform the exposure to treatment in real world and the effects of those treatment on the disease progression. So and capturing all the heterogeneity of the patients versus the treatments. So characterizing the responding profiles, capturing and characterizing also the adherence to treatments and so on into one common modeling and simulation platform that could really synthesize the evidence and inform the decisions through a cohort simulator that could again simulate what if scenarios for a decision, a given decision for a decision maker, be it from industry to position a treatment, or be it for a regulator to simulate a new treatment in the current therapeutic pathway, and therefore to estimate benefit risk over time versus the current standard of care. That's one. And really, my thinking is really that sort of integrative platform will enable also precision care, because then you can really inform also at patient level, even at the point of care, any kind of decision around diagnosis, around prognosis, around prescription, and really, of course, with a dedicated application on top of it, support the decision-making, the medical decision-making at the point of care. And of course, to make it possible, we need to validate, to qualify, to engage with regulatory bodies, to make it regulatory grade and validated for the dedicated use at the point of care. So I think I will stop here, and I just wanted to really introduce this disease-centric mindset and this integration of evidence for decision support beyond R&D or discovery. Thank you very much.
Thank you very much, Billy. I think we could now switch to Professor Christian Aumann, who will talk about simulating something for drug development. So thank you, Christian.
Thank you very much. I'm happy to talk about in silico trials. I hope going into presentation mode now. Well, we all know the usual terms: in vitro, on the bench; ex vivo, outside the body; in vivo, inside the body; and in silico means carried out in the computer. And it's increasingly used in the context of computer modeling and simulation. So an in silico trial refers to the use of individualized computer simulations in a cohort of patients during the development of a regulatory evaluation of a medicinal product, medical device, or any medical intervention. Currently, there are several projects funded by the European Commission dealing with this issue. The majority of them are dedicated to issues in cardiology. There's one stroke project ongoing. And we have in the US a big program called Model-Informed Drug Development. We are involved in one of these EU-funded projects. That's the SimCore project. And this project has the objective to establish a computational platform for in silico development, validation, and regulatory approval of cardiovascular implantable devices. There are the two devices involved. You can see in the left lower corner. And we have here really two main components in this project. One is a virtual core generation and validation, and the second is a simulation of the device effect. In the end, the platform will represent an open environment, and it will be usable by device manufacturers, researchers, medical authorities, and regulatory bodies. With respect to in silico trials, there are standards and guidelines available. In the majority in the US, we have a reporting guideline from the FDA. We have a model for assessing credibility of computational modeling through verification and validation. From the European Medicine Agency, we have a strategic reflection on this issue. We have a position paper from the Avican Alliance, and this organization is aiming together with other organizations at providing a good simulation practice, similar to good clinical practice of clinical trials. And then we have the HDR arena, which is also providing guidance for conduct and reporting of modeling and simulation studies. There are some, not many studies available and have been published in silico trials. I know that one of our participants, Kit, was also involved in such a trial. And there are also some very successful case studies have been performed to show the potential benefit of this kind of approach. So we have modeling and simulation for metallic implants in MRI scanners. We have applications looking at infection treatment and on feasibility of clinical trials. We have a modeling and simulation platform for tuberculosis with an immune system modulator. We have in silico disease models of chronic hepatitis. and CT-based models to predict the risk of hip fracture from engineering outputs, I would say. So these are only examples, but there are examples, and these kind of approaches have been used really in the exploration of medical devices and medicinal products. So what can be achieved if you apply in silico trials? Well, the target is to reduce the duration and sample size of real clinical trials, maybe to increase clinical efficacy by focusing on specific subpopulations, to improve patient safety, to increase the power of a trial, to optimize the methodological study design, or in trials on medicinal products, for example, pick up the adequate dose and select comparator in drug trials. And the last slide is how we approach that in the SimCore project. So the SimCore project starts with a model for the implantation for the disease and the implantation of the devices. Then there is a preclinical validation and verification phase. Thereafter, clinical trial planning starts, and then a virtual cohort is generated. This virtual cohort is validated, and if the model is adequate and validated, it can be used for prediction. If not, maybe additional human data have to be collected and integrated into the project. So we are quite happy, and we look forward how we can perform Dutch in silico trials in a specific statistical environment, and we have also clinical data, and we getting access to clinical trial data to do as much validation as possible. Thank you very much.
Thank you very much, Christian. I suggest we switch to Professor Jim Collins on a slightly different topic, but still much AI-based for clinical drug development. Thank you, Jim.
Let's see. So thanks, Raphael. So what I really want to tell you about is the Antibiotics AI project, which we recently launched at MIT. You know, as many of you can appreciate, that in the midst of the COVID pandemic, we're also facing another global crisis related to infectious diseases, and that's around antibiotic resistance. The number of resistant strains has been growing decade upon decade. This had largely been limited to hospitals, but now these resistant strains are out in our communities. Intriguingly, even in the midst of the COVID pandemic, antibiotic resistance and bacterial infections are playing a major role. 1 out of 7 patients who are hospitalized with COVID have a bacterial co-infection. 50% who die have a bacterial co-infection. This challenge is exacerbated by the fact that biotech and pharma have been getting out of the business because of economic constraints. Cost just as much to develop an antibiotic, but antibiotics so much cheaply than a blood pressure drug or a cancer drug, and administered typically acutely. What's lost on many is that the golden age of antibiotic discovery was in the 40s, 50s, and 60s. So before the biotech revolution, before the genomics revolution, before the AI revolution. And what we became intrigued on about 3 years ago, Regina Barzilay and Tommy Joklik, 2 of my AI colleagues at MIT, was to see, could we use AI, deep learning specifically, to address the antibiotic resistance crisis, the gap in antibiotic drug discovery. And so what we did to start was really in an unfunded project, put together a small training library of 2,500 compounds, 1,700 FDA-approved drugs, and 800 natural compounds. We applied these to E. coli and asked which of the molecules exhibited antibacterial activity, granulized it, just giving it a 0 if not antibacterial and a 1 if it exhibited antibacterial activity. We then took that phenotypic feature data along with the molecular structure information on each compound and trained a deep neural net, specifically a message passaging graphical neural network, that would look at each compound, bond by bond, substructure by substructure, scaffold by scaffold, associating it with antibacterial activity or not antibacterial activity. We then applied the trained model to the drug repurposing hub at the Broad, which consisted of about 6,100 molecules, and asked the model to identify, predict molecules that will make for good antibiotics, but don't look like existing antibiotics. Only one molecule fit those criteria. That's this molecule shown here called halicin. that turned out to be a remarkably potent novel antibiotic. It was effective against a broad range of multi-drug resistant, extensively drug resistant, pan-resistant bacterial pathogens, including TB. And we demonstrated its efficacy in animal models, both against Acinetobacter baumannii and C. diff. It acts through a novel mechanism of action where it disrupts the pH component of the PMF across the membrane, disrupting the ability of the bacterial pathogens to generate energy. We then retrained the model, then applied it to the Zinc database that includes 1.5 billion molecules. You could never empirically look at this many molecules. We looked at about 10% of the database. Only took about 3 days to screen with our models. Now identified hundreds of molecules that are predicted to make for good antibiotics, but don't look like existing antibiotics. We were able to purchase about 23 of these, 8 of which demonstrated antibacterial activity, 2 of which captured here demonstrate broad spectrum antibacterial activity. On the basis of this, we've now launched what we're calling the Antibiotics AI project at MIT. And we're now looking across 7 different pathogens, including TB and Klebsiella and Pseudomonas, collecting training data now with a much larger training library of about 39,000 to 40,000 molecules that can make for good antibiotics. We're now using the trained models to get after explainable AI, looking at chemical structures and mechanism of action for each of the predicted compounds. We are using the insights to both discover and design novel antibiotics. We're looking at combinations, and we're integrating these efforts with AlphaFold, for example, to get after both insights into antibiotic promiscuity and to lead and advance around target-based drug discovery. And so we're hopeful that integrating AI with these systems approaches, that we can actually gain an edge in the battle of our wits against the genes of these nasty pathogens and expand our arsenal against this growing threat of antibiotic resistance. Thank you.
Thank you very much, Jim, for this enlightening example. I now propose we switch to Dr. Hamzy Temani. Yeah. Thank you.
Sorry, can you see my screen?
Yeah.
Can you hear me?
Also.
Great, perfect. So actually, as I mentioned, this is a kind of a new initiative that started last year in France. And I wanted just to present the objective of the ambition for this initiative. So as you may know, like Johnson & Johnson has like a larger fingerprint in the healthcare and pharmaceutical industry. We're one of the top 50 Fortune Change the World list, and we are impacting around a billion patients around the world. So we work in collaboration with different entities, with academia, large pharma, venture capital, startup, government, and large biotech. And we have more than 140 active collaboration. Also, we have like a large fingerprint, worldwide fingerprint, and an investment of around 9.6 billion around the world. So within computational science, so what we're trying to do is really to leverage large-scale data set that is contained of different data sources, and the ultimate goal is to combine these different data set with also different methodology, with the ultimate goal to have a better target selection and de-risking. And as you may know, like for instance, we are one of the pharma member for the UK Biobank Consortium. We get like very large data set with kind of challenging way of analyzing the data, combining the data, and also leverage the computational facility. So nowadays, there's different challenges in how could we combine the data, how can we look at the quality of the data, how could we run hypotheses, how could we accelerate the running time to get from the raw data to the insight. And those are sometimes solved problem, but most of the time are still open challenges, especially when we scale up in the term of the patient. For instance, the UK Biobank is expected to generate different data type for up to 500K subject participating to the study. So within the AI and Genomics Initiative in France, we try to work internally with the Johnson Global R&D teams across the world. And we also collaborate with the Johnson & Johnson Innovation Center, and mainly with the London Innovation Center that is covering the EMEA region, and also with our colleague from Janssen France, with an ultimate goal really to connect with the French biomedical research ecosystem. And our target is really to look for talent, either by recruitment or by collaboration, scientific collaboration. We are also interested in identifying like interesting subset of data that could answer our research question. And we are also interested in different methodology, either to accelerate existing methods or to develop new method to tackle a complex and challenging problem. And ultimately, we're also interested in infrastructure, either computational, through the mean of accessing new technology like acceleration hardware, NVIDIA GPUs or quantum computing, but also technology infrastructure that allows to generate a new modality of data. So this is in a summary what is the aim of the initiatives. And last year, we've been able to have a first contract signed with the Pasteur Institute to look at the immunology, profiling and understanding of the different responses. And we are also one of the industrial partner of the Prairie AI Institute, with which also we're trying to address some of the challenges in AI. This is it for my part.
Thank you very much. And now we will hear Professor Kytrous. Yes.
Oh, let me.
And we see the slides.
Yeah. Well. Yeah. This will work, I hope. Yeah.
Yeah. Perfect.
Okay. Thanks. Thanks very much. And I think this is a very interesting winter school, and it's addressing topics that I will approach more from the clinical sides, both from a hospital perspective as well as my regulatory role at the drug regulatory agencies, because they are actually reaching the clinic and they are reaching regulatory decision making. I think that's really important. I want to highlight 3 points which are close to my heart, but I will do so through two initiatives that I'm actively involved in at the moment. And one is, I have to admit, also COVID disease. Very early on, there is lots of research also that we've been involved in very early on to uncover the nature of the disease, being able to also predict what happens to patients. And in the Netherlands, we identified quite early that should not happen in a dispersed session way. So each university hospital using its own data to do research, but actually we should share as quickly as possible. And the WHO also aims to facilitate that by very early on in the pandemic defining a key set of electronic CRFs to share data. And what we are doing in the Netherlands at the moment is use that as a template to share data from observational studies, but also electronic health records more generally across all the diseases. What we noticed, this was an ideal setting, we thought. We have a worldwide standard defined up front in terms of data. So this should be easy. And the thing that I want to highlight, it turned out not to. So even all the centers in the Netherlands were using the WHO templates and agreement. It was a large effort to truly standardize the data to be able to share. So that's and one of the key points that we also were well aware of, but learned again, is that the translation from data you routinely collect on patients to data that you can actually use to model and to more advanced to artificial intelligence or machine learning algorithms, that's a major step in terms of data quality. The data doesn't necessarily meet the quality standards that you're looking for. So it's a kind of lessons learned I would like to share, and it's a crucial part of any AI and machine learning effort, but I think we are learning fast, and we've used the pandemic to do so. So that's one key point. The other one, totally other extreme, because with COVID-19 we have very many patients very quickly, is the other extreme where we have an interesting project ongoing is in rare cancers. And as an example, salivary gland neoplasms, they are truly very rare. And they're also not all the same. So even within the very rare neoplasms of your salivary glands, it needs personalized medicine. They need to be treated differently. But to learn quickly here, you have small data sets, and they actually hamper building up your knowledge because they're essentially too small. And also here we see that this is more on a global scale. Each country might centralize, but then still the data sets are very small. And there's a high risk of actually having selection bias in those data sets. And currently, and I think that will be an experience shared by the other panel members as well, getting all data in one place is, I think it's actually technically not very difficult, but ethically, logistically, administratively, and regulatory, it's a huge challenge. So it's not, you might think, well, let's pool the data across the different countries, but that's not an easy route. So what we are doing at the moment is two things. First of all, we need to understand the disease, which is very rare in small data sets. And then you will notice that whatever complex algorithm you use, there is a risk of overfitting. And maybe you might find that not problematic for prediction, but actually to understand the disease, there will be serious bias. You might be wrong in the predictors. And that is quite independent of the type of complex algorithm you choose. That risk is everywhere. So what we are developing now is actually what you've been, I think, addressing this afternoon, being able to do the algorithms in a federated fashion, leave the data where they are, the algorithm travels around. And we are at the stage that we are able to provide algorithms to which we can actually prove they reach a similar efficiency as if you had the data together. And if we are able to do so, that will be a major accelerating factor in terms of learning across many databases. And the other route to explore is that we develop more powerful methods that actually deal with overfitting, that are able to avoid the problem, so that actually the conclusions that you do draw are unbiased in terms of the predictors that you identify. So those are, I think, important developments. So my key point was to point everybody to the real pressing need of quality and standardization of data. I think underestimated problem of overfitting in all the complex models that we use, and really promote the fact that we should develop federated learning and do the methodological development that's needed there, because I think that will truly accelerate much faster than trying to get all the data together. So I thank you for your attention, and happy to discuss.
Thank you, Kit. Thanks a lot to all of you for the presentations. I think we can move to some questions, and I would like to ask a first question. It's primarily for Jim Collins, but I would like also perhaps having the views of Billy Amzad and Hamzy Timani on that. Jim, we've talked a lot about data, and what type of data sets do you think would be needed to enhance the impact of AI in drug discovery?
Yeah, it's an excellent point. You know, my sense is multifold here. One is, I think the groups that make the commitment to collect their own data with the modelers in the room as we design the studies, the AI experts, are the ones who are going to win, meaning they'll have the great value. I think we're at a stage right now in the next decade or so, the groups that can most smartly integrate machine intelligence with human intelligence will be the strongest. Specifically to data, the next comment is that I don't think we're yet really in the big data phase in biology, medicine, and biotech, with the exception of sequencing. And again, I think it's largely from the fact that most groups aren't making the commitments that are needed. in order to collect those data sets. There's been very nice efforts on trying to cull and put together outside groups, but even picking up on Kit's excellent point at the end, it's hard to standardize across these different data sets. And thus it becomes very challenging to integrate them in a meaningful way. So I think in drug discovery, it really will be, can we get after diverse chemical perturbations in the appropriate cell lines with multiple readouts, meaning getting after multiple phenotypic outputs, whether it's gene expression, protein, metabolites, phenotypic, such as impacting cell growth or cell death, permeability, and coming now at this from a multidimensional standpoint. So again, I think data are king. And intriguing, I think AI, look, we have many world's experts in AI. We have a lot of interesting challenges there around using smaller data sets, given that it's going to be a while before we get to these larger ones.
Thank you. Perhaps, Billy, what will you say to that?
Yeah, I fully concur with what has been said. I may add some one idea on real-world data for discovery, whether they are linked or not to omics or biodata. I think in order to anticipate the impact of the discovery and to prioritize discovery programs, those real-world data that inform, they are massive. They are surely carrying a lot of information on mechanism of actions, on heterogeneity of patients, and so on and so forth. So capturing this information and this heterogeneity upfront in the discovery phase is, I think, key for successful and impactful AI analytics and AI-driven drug discovery.
And Ramzi, you now work in a big pharmaceutical company, but you also have a bioinformatics background, so, and you work in genomics. What would be your take on that?
So maybe I'll have another angle to the question, because basically from an abstract perspective, so if you add data and another complementary data, you get a better result. However, if you add data and noisy data, you might perturb the signal. And if you add noise to data, you make it worse. So the question, and I'll get back to the first point, is like, how can we make sure that we define with the knowledge expert that we collect the right data? And once we collect the right data and we are clear about the type of data that has to be combined, the second challenge is how can we ensure that the quality of the collected data is good and that we go through the proper quality metrics to do that. For sure, there's a lot of paper that show different kind of omics combination. I worked more on that area where we see that by combining subset of data in the omics field, we get a better accuracy. However, now that we have a kind of multi-layer data with different data type, I believe the most important thing is really to start from the proper research question and look at the literature to see what combination we can use. And I think today, the good thing is we have the proper computational power to run multiple hypotheses, do multiple testing in parallel. But ultimately, those models are valuable once they are validated by the end user expert from the biology question, the one who asked the real biology question. So I think the most important thing is not how much data we collect, is like how we collect it and how we make sure that the data we get is of good quality and that we have enough sample to have like a good statistical power in assessing the results.
Okay, so while you are talking about quality of data, this reminds me that we could also discuss about the potential concerns on the quality of routinely collected data and how those could be taken into account when one does artificial intelligence, machine learning, statistical learning. And this one would be more perhaps for Kit, but I would still value the input of Billy and Jim after that. What are the concerns or the limitation on the quality of data?
I think that's a good question. It depends a bit on the source of the data, and I will give two examples. If you think about electronic health records and the data, that there's a huge amount of data, but that's not collected with the purpose of doing research. So that there are serious quality concerns there. And I think one of the key points there is actually to bring research results and decision maker closer to the clinicians that actually are responsible for the data, that they get a feedback loop, that what they collect in their systems is actually useful in their decision making. If we get that feedback loop closed, I think actually the data quality will improve as well, because it will matter to their decision. So I think that's one crucial element. If I take another example, also from my experience early on, organoid research. If you have different organoid labs and you want to standardize them, actually the whole effort of collaborating and being willing to share the data across different stakeholders is an incentive to increase the quality and increase your standardization. So also there you might think, well, it's lab, it's easy. No, it's not. You still have to investigate differences and being willing to share and collaborate is actually an incentive to improve the quality on all the participating sites.
Thank you. Perhaps, Jim, on the issues about quality, we've talked about quantity, but also what inside the data.
Yeah, so I'll give a couple comments. One, I thought, excellent points around is that, the challenge is as we move toward biology versus say just sequencing a genome is that biology is highly variable. The young kid on my benches to the left will get data very different from the one on the right. And it's that it's hard to standardize across biology when you get after the dynamic feature, and even within a lab. And so then it becomes the challenge, to Ramzi's point, on the stochasticity that's frankly intrinsic to the living cell. And so I think regarding that, I think we need to move more so also toward capturing uncertainty in our models and getting after Bayesian approaches that can characterize how certain are we in the predictions that are coming out of the models. And I haven't seen that as extensively used. We haven't used it as much in my group, but we're collaborating with groups now at University of Cambridge, which is really one of the center points for Bayesian approaches. And I think that's going to be needed given, again, both the points that Kit alluded to on quality across the labs, but also frankly, just to the intrinsic stochasticity of the systems we're studying.
Thank you.
Please,
Billy, yes.
So, as a Bayesian statistician, I can only concur with what Jim was highlighting. And maybe one comment maybe on real world data as well, in terms of quality and in terms of what we mean with quality here. Of course, for discovery, real world data have not been collected for the purpose of making a discovery or clinical trials. So here, you of course, quality means that you have a lot of bias. You may have a selection bias. You have missing data, and so on. But you have heterogeneity. So there, of course, you need to be able to handle those gaps. The size of the data won't be enough to tackle the issue. You need to maybe to benchmark. You need to have specific data to address those biases, and to combine also with RCT data as well. Again, integration of data. Now, what I want to emphasize on is that what makes real world data low quality for discovery is actually what carries a lot of information for payers or for maybe public health decision makers, because if a patient has missing data, if patients are routed or are selected by physician, by prescribers one way versus another, it means a lot on that there is a hidden reason or good reasons why you don't have a data record in your data set. So this information is crucial for payers and for public health decision makers, and this is also the challenge for AI-driven healthcare to capture this information.
Rafael, if I may comment, I think the way I see quality of the data and the quality control measure is, I think it's a continuous journey. In my previous role, leading the bioinformatics core at Sidra Medicine in Qatar, we've been in charge of doing the quality control for the Qatar Genome Program. And what you notice is it's a continuous process where each time you realize there's an outlier or looks like an outlier, you learn and you improve your quality, recommendation. because sometimes the data coming from the sequencer has some standard metrics, but even those would miss some details. You do the mapping, you realize there's some other outlier, and then you do the variant calling, and then you see some other result that escaped the first quality control. So what happened is really it's a journey where you improve the quality of your checks along the way. And as far as I recall, there's still no best practice for genomics data. I've seen one for RNA-seq a couple of years back, giving some recommendation, but we don't have one fit for all way of doing the QC for all the data. So I think it's still, again, driven by the kind of question you want to answer. And even for the filtering of the data, that is also related to the stringency of your question. When you do it for research purpose would be different from when you do it for clinical diagnosis at the hospital. So I think it's very variable and depend on the context.
Thank you. Ramzi, I think I have a question still for you, but I would like to have the input of Christian and Kit on that question also. But we have talked a lot about data, but we also saw on your slides that it's not one type of data. You have to integrate, and we have heard the term data integration. You have omics data, you can have imaging, but Kit talked about electronic health records. How can you best leverage all those heterogeneous data for arriving at something for precision medicine or even healthcare in general?
So that's a great and complex question to answer. I spent like my PhD trying to work on data integration. And basically from conceptual point of view, there's different way of integrating the data. So basically you have what we call the early integration, the intermediate integration, and the late integration for the eye. So basically the early integration is trying to convert the data, make them as one single source of data and create a model out of that. The intermediate integration will take each data separately, will create kind of a matrix, will aggregate the matrix, and based on that matrix, will create a model. And the late integration is a method that will take each data separately, create a model, and let the model vote. So here again, it's a matter of what we want from integrating the data, because we could use the data to be integrated without even needing the AI or build a model. We could create a dashboard to have a better visibility about the information of the patient at the moment T, because that would be also valuable information. So the way we integrate it, how we create the dashboard, what data should come in when we call a certain type of data, because we know that adds value. And we got back to the paradigm value versus information versus noise. So you don't want to have like a very filled screen for the doctor with a lot of information while he only needs 2 or 3 information. And I think, again, we go back to it. The critical point is like this interaction between the end user and the data scientist working together, trying to understand the problem and trying to identify the full solution to answer that problem.
Oh, thank you. Perhaps with data integration, at some point, I was thinking that also you may want to train algorithms separately, and then at some point to combine things. And this reminded me that Kit told us about federated learning. Perhaps you want to develop more on that, or?
Yeah, maybe for your initial question, my thinking was more that I really would, in terms of data integration, put the research question pretty central, because that might also determine which level of aggregation you actually need. You might not need for all research questions the same level of aggregation to integrate the data. So thinking in terms of research question and your smallest experimental unit, if you like, I think that's helpful in those decisions. So I'm kind of wary of general data integration and getting everything together. You would want to know which questions you actually want to address. And I think federated thinking might, let's say, prevent you from making it technically too difficult in actual physically getting data together, because I think we've passed the point that will be possible. I think we will not easily have all data together. I think we're actually, in all initiatives that I'm aware of, data will not necessarily be in one database. We need to be able to be more flexible there. And that, for me, that very much depends on the research question you want to address.
And Christian, I thought that those issues might be also related to how you build your in silico trials, but also how your in silico trials contribute to the whole?
Yeah, I think that first of all, I want to say that I'm also in favor of the federated models, federated queries, federated analysis, because we learned from the implementation of the GDPR, which should have become harmonized throughout the EU. It's not the case. It's national, it's regional, so it's very, very difficult to move data to a central place. So the future will certainly be for federated approaches, I think. Another problem I see at the moment is that that we have different worlds. We have the medical care world, the healthcare world, and the standards used there, if standards are used, and we have the research world. But it's meanwhile, I think there are some developments which make me a bit optimistic. That is, researchers now try to bridge the research standards like CDISC for clinical trials with standards they are applying for electronic health record data like OMOP, HL7 FHIR, and all these kind of things. So there will be some progress in the future, but as it was discussed, everything is dependent also on the quality of the data. You can bridge and link what you want, but if it's not the quality, then the result is disappointing. But anyway, there is progress in this field.
Thank you. And Christian, while we are talking about that, I would like to come back to the in silico trials.
Yeah, okay.
You've showed us the infrastructure you're trying to build, and there are clearly works on the domain. But then one question might be asked by end users or regulatory. So perhaps Kit might comment on that also from his other job. But how could we set the role of in silico trials in relationship to the real clinical trials? Is there something like a timeline between both, or not so much?
Yeah, I see a supporting role of in silico trials, a supporting role in the sense that it may help to optimize trial design. And there are some nice examples showing that you can reduce the duration of a trial, you can find a better endpoint, a clinical endpoint, you can decrease sample size, you may increase the power of the trial, you may take a better design, a methodological design. So all these developments may help in the end to reduce costs and time to market for medicinal products and medical devices. So I see the in silico trial integrated in the process to improve the clinical trials. That's the main point, I think. It's not except for very, very rare cases, it cannot be a replacement. There must be a validation in the end, I think.
Thank you. Billy, you wanted to say something on that.
There's just 2 quick points. One is that I fully concur with what has been said. I can augment it with saying that you can also consider in silico real world simulation, so in silico cohort simulation to even better optimize trial design, in the sense that you can align the design and the population, the target population, for example, to the expected real world value. It's one thing to get the approval, but it's another thing to get, again, the drug prescribed and have an impact in the real world. And for that, you need to go a little bit beyond the authorization and look how one drug would impact in the real world, basically in the treatment sequence, for example. That's one thing. And second thing is that it also just specifically for in silico trials, you may also be able to, when you have data already on, for example, on the control arm, on standard of care, you can use it to basically build a historical control that you can simulate and use as a reference, and that have, I think, a lot of precedents.
Yes.
Precedents. Yeah.
Perhaps Keith or Jim on that topic.
Yeah, maybe I think yes. I think we are quite aligned here. It's supportive, but we should also not, let's say, my 2 hats. So regulatory, you would want to see the clinical validation, and I think that's really important. but there are also questions which are actually quite difficult to address, could be helped by in silico trials, and that's translations. I think the example was mentioned from clinical trial settings to real world environments. Would you need another trial, or could you actually have in silico trials to see what would happen? And the same is, for instance, and I think there's also more openness to modeling and simulation approaches, translating results in adults to children, for instance. So there are a couple of areas where there is direct added value of in silico trials.
And perhaps for Jim, at the other part of the development, because you are in the drug discovery, would you go for also in silico testing of the AI found antibiotics, for instance, or do you think first it would be more in back and forth between AI and discovery, then clinical testing, and then perhaps modeling, and then?
I think that we have found that the most efficient is to do the back and forth, to really get to a reasonable and accurate predictive model. Then I think it's valuable to apply the model to these much larger spaces that I alluded to that you empirically could not screen. But I think we're still at a stage where you got to experimentally validate what the prediction is. So I wouldn't feel comfortable only going with the in silico prediction into a human. I would want to validate it in vitro, ex vivo, mouse work, non-human primate, advance it. But the modeling significantly cuts down on the experiments I need to do in my exploratory phase and allows me to explore a much larger chemical space, say in my world, that just would be impossible otherwise.
Thank you. Unfortunately, the roundtable is moving to its end, but still not. Before we take questions from the audience, if there are, so I urge the audience not to be shy and to ask questions in the chat, so we can query our panelists on that. I will have a last question. I think it's for Billy primarily, but Christian and Ramzi would be welcome to answer too, and anyone in the panel, of course. How do you think we can switch from many analyses that are done often we use AI for proof of concept and many small scale analyses to some extent, to something which is more integrated, not in terms of data integration, but in terms of reflection, more integrated and collaborative to try to answer questions, but also how can this impact the business and medical decisions? So really the end users,
yeah,
and not only the people doing AI or research.
Thank you. That's a good question, and it was in line with what I was trying to sketch out. Going beyond R&D and looking how how all those AI methodologies and those data-driven approaches can be combined together to ultimately support integrate evidence and support decision making for physicians at the point of care, for epidemiologists driving vaccination policies, for example, and industry leaders that have to prioritize programs and so on. And for that, so that's why I was kind of, I think the disease-centric mindset, the disease-centric approach is key in that. And disease-centric, I mean also patient-centric, because here we're talking about one patient with his disease and trying to kind of inform, for that specific patient how a disease may progress and how a care pathway may be more efficient for this patient. So really this patient-centric and disease-centric approach, combining multiple data sources and of course multiple AI methodologies or algorithms, should be the only way forward. And what I was describing, actually, it may look a little bit conceptual, but actually we have, I have seen many examples in specific areas in neuroscience, in Alzheimer, in cancer, in cardiovascular diseases, where this was applied specifically and could really support, jointly public and private decision makers. And I think that's also the way forward. The future is really to have a more public-private approach to this evidence generation and evidence integration, because again, understanding the disease, understanding how one treatment can impact the fate of a disease is beyond one industry or one regulatory body. Is really it should be a joint effort. So that's why I think I'm promoting a consortium and public-private approach to the building of reference disease models, basically.
Hamzy, perhaps, or Christian, sorry.
Yeah. If I may make a comment, I think what is very important is to increase transparency and repeatability of the analysis, and that can best be served with open approaches. So I think we need platforms, we need archives where these methods, these applications, these algorithms are stored. And in order to be useful, I think we should work with standardized workflows, which are already available. And if you have these chunks of algorithms, you can then come into a situation where researchers can assess the material. They can look at a specific step of workflow they need help for. They can use what is available. They can integrate it with their application, and then you get much more out of what is available. So I think openness and standardized workflow, and very important, specific defined metadata for discovery is very important, and that's something where I think the AI scenery must do more to provide real, very informative metadata that can be used by other researchers, so they can really find what they need. So one could do a lot, I think, to improve here.
Thank you. So perhaps a last word to Hamzy.
Yeah, I think it goes without saying that today's problems are so complex that one entity alone could solve it by themselves. And it's important to really create this ecosystem where you have different teams from different backgrounds, from different fields, working together, trying to address this challenging problem. And I think this is in line with the philosophy behind creating the AI and genomics initiatives within the French ecosystem. And we've been working with the ARIS, for instance, with the other player within the pharma industry, trying to work together, try to solve problem. I'm also involved with the Choose Paris region, with the Club of Leadership in AI, to work also with other AI leader in other field, because sometimes we have the solution out of the box coming from another field. So it's very important to have this ecosystem at the national level and eventually at larger scale through, for instance, the AMI project, and Johnson is participating to a lot of the European project where public and private work together, trying to federate, I would say, their effort and energy to address complex problem, to help solve a problem and benefit to the patient. And I think it's very important to get that in mind.
Thank you very much. I'm afraid, but no, it's 1 hour. So even if I had negotiated some more, we are still exactly on the schedule.
You can use 10 minutes more. You negotiated that, Raphael. So please.
No, but there seems to be no question in the chat. So perhaps the audience is shy, or it isn't working for a reason I don't know. If there are no questions, I think I will try to have just some words to summarize what we've seen. And if our audience comprises a lot of young people working or studying in the AI domain, what I've heard is that clearly there is a room for AI in healthcare, or in the domain we tackle, which is more about interventions and drug development tonight. From the very, very beginning, and the amazing work of Professor Collins. So from the beginning of drug development, which is drug discovery, even the screening the molecules, to much, much later phases where we have heard the what we can do with analytics to identify which treatment would be better suited for which individual's patient characteristics, or post-marketing studies or evaluation. And so through all that pipeline, there are things to do, both in terms of application, but also in terms of development and research. And we've heard from Kit Roos that clearly there are topics that still need development. So I think it's good news for the younger one who wants to work on the field. We have, okay, so now I have a question. I will just end my sentence and ask you the question. Then the good news also, I've heard that some groups, and Ramzi told us, are looking for talents. So we have private companies, public funded bodies like ECRIN, universities. So many job opportunities, I would say, for the trainees, and that's good news. I have the impression that the ecosystem exists, but perhaps is in need or is organizing itself. So it's a matter of how it's up to us to organize things to get it right. And that's what I wanted to say. So one question is that, to what extent and at which conditions do you foresee the acceptability of these complementary AI studies by agencies, HTAs, and physicians? I'd say that it's primarily for kids, but I know that Billy or Ramzi have also experience with interacting with the regulatory. So, but Kit, I would say.
Yeah, I'm happy to give a first response here. I think what's good, this is a big leap. We have acceptability of a fairly broad field, but I think what we do know, and the EMA is heavily investing in understanding this field a lot better than they do today, with the big data task force and far-going initiatives to actually have data available and invest also in their own knowledge on artificial intelligence. So I think acceptability is a big leap, but there is understanding that this will actually change the field, and we need to know how that can impact decision making positively. So I'm fairly optimistic that initiatives to discuss approaches that are based on AI and improve decision making, improve our understanding of diseases and benefit risk assessment that we need to do, will be acceptable. But of course, the human is a very complex organism. So I think we will not quickly do without clinical proof of things that happen to humans.
Billy or Ramzi, I don't know who wants to answer that. So thanks.
Quickly, maybe, yeah. So indeed, so there has been more and more, successful pilots, successful cases. Now the real question is to switch from, again, multiple pilots or cases into, the accepted and endorsed by HTAs or regulatory bodies, and that's in progress. I think Kit mentioned the big data task force at European level. There are similar work streams in the US as well. So it is progressing. I think one key element is to have standards, as also Christian was highlighting. Not only standards on the data format and data quality and so on, but also on methodologies. We need some interpretable AI, or at least clinically interpretable AI. That's I think a major element, a key element that would enable this acceptance at the large scale.
Thank you.
Raphael, I'm going to make a quick remark, because I think maybe we are looking at the problem also a bit classically. We have different parties, and one has to accept or endorse other stuff. I think that will be moving as well. I think from a regulatory point of view, we are trying to do that partly together, and we actually need AI, machine learning, real-world evidence experts who are willing to actually look at the problems with a regulatory point of view. But we need to do that a bit more together.
Yeah. So if I may comment, I think that the challenge is like the regulatory body and the scientists speak different language. And to a certain extent, we should know better each other and learn the other language. And the major challenge is really finding the right spot between utility and privacy in the data. And if I may recommend, there's a World Health Organization report on the ethics and the governance of artificial intelligence in health. If you want, I could post the link. It's a 156-page report, but it's really worth reading to see the different perspective. And again, in healthcare is different from other area where prediction could be different. So if your prediction of your Uber time is late by 5 minutes, it's not as serious as having a prediction, a prognostic. So we take things more seriously in the healthcare field. Thank you. I think we are almost over, but there is a question by Katrina, which is not an easy question, I would say. I wouldn't like to have to answer to it, but it's for you and not for me. And the question is, how are the changes in this social technical work system of clinicians or healthcare professionals included in the developmental research of AI applications? Mm-hmm. And I think perhaps Billy could try to answer that, or Hamzy. Well, maybe I can take my own experience during the PhD. I'm from computer science background, and one day we find ourselves in Hotel Dieu working directly with clinicians. We spent almost 6 months trying to be nice to each other because we're speaking different language and there's nothing in place to let us understand each other. So what happened is, like, after a certain while, we started to complain that there's nothing happening because, you know, like we have like this weekly meeting and nobody is getting what the other is saying. So we put in place a process trying to, each time that the bioinformatician is presenting, he grab a biologist, go through the presentation and make sure that they get to a level where the biologist can understanding, and vice versa. So the moment you get that understanding between the two teams, we started seeing some collaboration starting to sparkle, and people see that we are always tackling the same problem from different angle, trying to help the patient, but that require somehow a dual competency programs within the university to help the doctor being educated in AI and help the AI scientists also being educated in the different fields and not just taking the data as abstract data.
Yeah, I have a similar experience working, developing, you know, analytical tools, decision tools for healthcare professionals or for regulatory decision makers. The AI specialists need to understand, need to be also part of those, you know, regulation processes, but also vice versa. When we are, you know, building a tool and that needs to be scaled up, you need also to include in the development process upfront the end user, be it a clinician or a regulator or a payer. And that's again, you know, lead us back to you know, the idea of really consortia, public-private approaches to these reference analytical technologies for decision making.
Thank you. I think it's the last word. Before leaving the floor to Emmanuel, I would like to heartfully thank our panelists for the presentation, their time, and the excellent discussions we have. So thank you very, very much. And the roundtable is now over.